AI, fungi, and the future of drugs. Sounds a bit like a sci-fi movie title, right? But what we're going to explore today is very, very real. It's about how these two seemingly separate worlds, the digital brain of artificial intelligence and the ancient hidden kingdom of fungi, are colliding to maybe, just maybe, kickstart the next golden age of medicine.
So let's set the stage, because this is important. For nearly a century now, we've been living in what you could call the age of miracle drugs. Antibiotics, all that stuff. But that golden age, well, it's facing a serious crisis. Our most trusted medicines are losing their punch against these rapidly evolving superbugs, bacteria and fungi.
And this isn't some far-off threat. It is very real. Let me just give you one number to think about. 1.7 million. That's the rough number of deaths every single year just from invasive fungal diseases. Just fungi. So yeah, this is a massive and frankly a growing global health crisis.
That's a pretty daunting problem, right? But what if the answer isn't something we have to invent from scratch in some high-tech lab? What if a solution has been hiding under our feet, in plain sight, this whole time, just waiting for us to find the right key?
Well, that key, it turns out, might just be fungi. And this is not some radical new idea. For centuries, we've been stumbling upon some of our most powerful medicines in this sprawling, kind of secret kingdom of nature.
And the track record is just incredible. I mean, think about it. 1928, penicillin from a simple mold kicks off the entire antibiotic revolution. Then, you jump to the 70s. Fungi give us statins, which have saved countless lives from heart disease. But this isn't ancient history. The pace is actually accelerating. Get this, 907 new fungal compounds were cataloged just this year alone. That tells you this treasure chest is far from empty.
Okay, so how are we suddenly finding all these new molecules? What changed? The secret is a fundamental shift in strategy. We're no longer just looking at the chemicals fungi happen to produce in a lab. We're learning to go straight to the source and read their genetic recipe book directly.
And the core of that recipe book is something called a biosynthetic gene cluster, or a BGC for short. The best way to think of a BGC is like a tiny molecular factory, and the blueprint is encoded right in the DNA. It's a set of instructions where each gene is like a different station on an assembly line, all working together to build one specific, complex molecule.
And the process itself is beautifully simple, really. The DNA holds the BGC recipe. The cell's internal machinery reads that recipe, and boom, out comes a unique and often bioactive molecule, a potential new drug.
But here's the crucial part, the real mind bender. Scientists estimate that the vast majority of these genetic factories in nature are silent. They just don't turn on under normal lab conditions, which means we've never even seen the molecules they're capable of making. It's this massive, invisible library of potential drugs, and we're just now figuring out how to unlock it. And this is where artificial intelligence comes in and completely changes the game. We are now developing AI that can read the DNA directly, decoding this chemical language of nature, and predicting what molecules these silent gene clusters are designed to produce.
And this really shows you the shift perfectly. The old way, traditional discovery, it was slow, it was manual, a ton of trial and error. You had to grow the fungus and just hope it made something interesting. The new AI-powered approach, it's fast, it's predictive. It looks right at the DNA and tells scientists what's possible, not just what's happening to be active in a petri dish.
So you might be wondering, how does this AI actually work? Well, the big breakthrough, the real game changer, came when scientists started treating the genetic code not just as a set of instructions, but as a language.
Here's how it works. The AI uses transformer models. Yep, the same kind of technology that powers tools like ChatGPT. It looks at a BGC, and it sees the functional parts of the genes, these things called protein domains, as individual words. The entire BGC then becomes a genetic sentence. So by analyzing millions of these sentences from all across nature, the AI learns the grammar, the rules of how these genetic words combine. And that lets it predict the meaning of the sentence, which is the final molecule it produces.
And this isn't just theory. There was a recent case study with a bacterial molecule called cyclooctafetin, and it shows how this tech is moving us beyond just finding new molecules and into the world of actually designing them.
So in this study, the researchers trained two different AIs. The first one was trained only on the BGCs we already knew about, our existing, pretty limited recipe book. But the second AI was trained on entire bacterial genomes, a much, much richer dataset. So when both AIs looked at the BGC for cyclooctanin, the first model predicted the expected stuff. But the genome-trained model, the one that had learned a richer language, it predicted that you could add several new and unexpected genetic words to that sentence. So they put the AI's prediction to the test. The scientists physically added the new gene, the new word the AI suggested, into the cyclooctatin BGC. Then they put this brand new, edited genetic recipe into a host microbe to see what it would make. The result was just stunning. The microbe produced a totally new derivative of cyclooctadian, a molecule that, as far as we know, has never existed before in nature. The AI didn't just find something, it helped scientists design something completely new. Now, this is incredibly powerful stuff, but there's a catch. While this approach is making huge progress with bacteria, fungi are a much bigger challenge. And it all comes down to the AI's data diet. When you look at the prediction accuracy, the difference is stark. You can see right here, models trained on bacterial data are hitting accuracy rates around 80%. Pretty good. But for models trained on fungal data, that number drops significantly, down to around 60%, or sometimes even lower.
So what's going on here? Is fungal DNA just harder for the AI to read? Not at all. The problem isn't the AI itself, it's the limited and really biased data we're feeding it.
You see, our library of known fungal BGCs is incredibly biased. This chart really shows it all. The vast majority of our entire training data comes from just two genera, Aspergillus and Penicillium. It's like trying to teach an AI all of human literature by only letting it read Shakespeare and Dickens. Yeah, it'll become an expert on those two, but it's going to struggle with everything else. This lack of diversity severely limits the AI's ability to learn the full language of fungal chemistry.
But even with this data challenge, the path forward is clear. We are standing at the beginning of a true revolution, one that moves us toward a future where we can not only read, but also write the language of biology.
And the implications go so far beyond just medicine. Imagine AI-designed drugs that can finally defeat those resistant superbugs, or new targeted agricultural products from fungi to protect our food supply. We could even engineer new sustainable materials. We are truly entering a whole new era of discovery, powered by our new ability to decode nature's chemical secrets.
And that brings us to the final, crucial question. We have only just started teaching AI the language of life, and it's already helping us write sentences that nature never has. So as we get better at this, and as we expand its vocabulary with more and more diverse data from the fungal kingdom, you have to wonder, what incredible cures and solutions are just sitting there in the DNA, waiting to be read?